Thank you very much to the organisers for the opportunity to present at this year's BGRI Technical Workshop. In my presentation today, I'm going to share with you some of the solutions for faster genetic gains and improved wheat seed delivery that we're implementing within the Global Wheat Programme here at CIMMYT. I also wanted to acknowledge a number of colleagues, including the breeding team, our scaling and socioeconomics experts for their contribution to this presentation. So, addressing supply and demand. We have many evidences of the impact of the CIMMYT wheat breeding program in supply of germplasm to partners around the world. This is a long history, over 50 years of delivering improved germplasm with evidence for the global reach and impact of this. On the supply side, we're asking the question, can we do more? Can we speed up the breeding process? Can we reduce the time it takes to get a variety into the hands of farmers? Can we improve the accuracy of the breeding process and the cost conversion in terms of delivery of genetic gains? Can we integrate new knowledge and innovation that comes from pure science research into application? Can we apply it into practical breeding? And can we better tailor our supply? So can we develop more structured linkages to national breeding in order to provide more targeted, tailored material to meet the local needs of our partners? We're also very interested in demand side. And this is because although we've supplied huge amounts of improved germplasm, we still see persistent issues of low uptake and slow varietal turnover. And if we're to achieve our ultimate aim of increasing genetic gain in farmers' fields and thereby really impacting the livelihoods of smallholder farmers, we need to address not only the supply of better material, but also the reasons why it's adopted and the reason why it's grown and preferred by farmers. And there are many questions underlying this, which I'll highlight today. Can we more clearly address our market demands, a missing step between the registration of a variety and its widespread adoption? Can seed distribution methods and models be enhanced? Can we use learnings from other crops or other systems or other industries to make these distribution methods work better for the cereal seeds? And how do we incorporate consumer and market preference as well as the gender dimension? So really looking at how we can address and optimize both the supply and demand components going forward. And this will ensure we have impact at scale, really about providing material and making sure that material gets onto farm and into the hands of the people who will use it. So our overall aim is to accelerate genetic gains from breeding, as well as to support public and private sector efforts to get more improved germplasm into farmers' fields in less time, thereby boosting our impacts on livelihoods. So just to set the context for this presentation, as I've mentioned, the CIMMYT Global Wheat Programme has a long history of achieving large impact through provision of improved germplasm. And this comes from a centralized breeding program run here in Mexico, a shuttling material between central and northern Mexico, allowing selection for multiple disease stresses, as well as for yield potential and stability of performance. It also includes the Kenya shuttle, which allows us to screen intensively for stem rust, particularly as well as for yellow rust. And the results of this centralized breeding program are shown in the graph on the right-hand side, which shows the contribution of CGIR material, both as direct releases and of parents in the parts of the world where our program seeks to deliver impact. We can look at supply of continual grain yield progress over time, both in our simulated selection environments in Ciudad Obregón, and the graph on the left shows a recent analysis of the breeding progress over time. And this shows in both the optimal environments, shown by the green line at the top, and our drought and heat-stressed environments, shown in the bottom two lines, that we see these increases in grain yields delivered by the breeding program over time. And this shows we can continue to increase the grain yield potential and make that available to end users. We can also look in our target environments, and on the right is an example of a recent analysis from the Indian target populations of environments where our material is grown. And this again shows an upward trend in productivity, in productivity and grain yields over time. So we see we can continually and have in the past and can continue in the future to supply continual yield improvement. We also see at the same time a supply of new tools and technologies which supports our progress towards the objectives of increasing genetic gain. And we can see a range of technologies, many of which have already been highlighted in the forms of genetic mapping, genotyping, phenotyping technologies, better identification of material, the ability to test it at scale, markers which are now available. And really one of the challenges is working out how to practically apply this supply of new tools and technologies into our breeding pipelines. So this brings me on to the Accelerating Genetic Gains in Wheat project, which I'll describe now. And this is really looking at the earliest stages of the breeding program with the objective to reduce breeding cycle time. So the core part of this project, as the title suggests, is the acceleration of the breeding process. And we're achieving this in a number of ways. Firstly, through faster breeding. And we've invested in a large greenhouse infrastructure in Toluca, which is being used to reduce the breeding cycle time from 6 to around 3 years per cycle. We're also using genomic selection for the rapid recycling of parents, as well as speed breeding for novel trait introgression for the development of parents or the augmentation of existing breeding products with genetic loci of interest. So this is a view of this greenhouse facility in Toluca, which is now operational, and the first cycle of crossing is now complete within the facility. And this is a very large area of covered screenhouse and allows us to move through the generations more quickly than if we were working in the field as previously. So you can see here around 1 hectare of accelerated bulk generation advanced material, the earliest stages of the breeding program. And this is Suchasmita Mandal here, who is leading the optimization of these rapid schemes, which require a huge amount of logistical, practical and technical input. And you can see on the right the recent round of crossing, which is being completed. Around 1,500 crosses are now made and harvested within this facility. I also wanted to mention the resistance breeding strategy in the context of this meeting and say that all of these accelerated schemes need to consider the necessity of providing a durable rust resistance in all of the germplasm coming from the program. And this is a slide from Ravi Singh, which really shows the rationale behind using adult plant resistances and really monitoring and carefully deploying the available genetics in a sensible way. And the graph on the right shows you the impact of having 4 or 5 of these minor genes in comparison to just 1 or 2 or a susceptible variety. And in our context as CIMMYTs, the resistance package that we provide is the first line of defense for many of the farmers who grow wheat in the geographies that we serve. And this APR-based resistance breeding strategy is underpinned by international disease phenotyping, which is hugely important not only for the rust, but also for many other diseases, as well as for a number of the stresses to allow us to really test and reliably provide genetics, which is able to withstand many of the challenges that we've seen in the past or we see emerging in the future. And here is marked on this chart that the yellow rust, leaf rust and stem rust phenotyping capabilities that we have, which are hugely important for ensuring the material has a robust and durable resistance. So in terms of how we're progressing this within our accelerated breeding scheme, one of the areas of activity is integrating multiple race-specific resistance genes through our speed breeding platform. So this speed breeding platform also exists in Toluca, where our greenhouse facility is based. And you can see here we've converted a part of the facility to be used for this purpose. And here, where selectable markers are available, they're being integrated via speed breeding trait augmentation. So taking elite backgrounds and rapidly introgressing these loci or multiple QTL or loci via speed breeding. So in the first cohort, which was complete in December, January, there was 140 crosses, achieving around 3 months per crossing cycle. And these have subsequently progressed through further generations. The second cohort of parents was initiated in June, with 70 crosses progressing through the second cohort of material. And just to highlight here, I think one of the bottlenecks for us is the reliability of markers, particularly for some of the disease resistance loci that we want to make available within elite parents. And this is one of the current limitations of this system, the ability to very accurately use marker-assisted selection within the speed breeding system. Just to give you an idea of the current material progressing through our speed breeding line augmentation. So here we're really aiming to produce parents which capture all of these different genes or loci of interest, and these parents can then enter the breeding pipelines. And you can see here summarized the current targets for both red wheat as well as durum wheat. And in this context, highlighting the stem and yellow rust resistances, which are currently being introgressed and pyramided, as well as other disease resistances, aphid resistance, drought and heat tolerance, as well as both a disease resistance and a grain weight locus in durum wheat. And what we're trying to work towards is really a much clearer framework and pathway for deployment of trait associations. So we know in the literature there are many reported trait associations. We know that from the work at CIMMYT and the work of many others. And really to look at where each of these discoveries fits and how it can be deployed into the stages of the breeding process. So if we start in the top left-hand corner, we have a large effect QTL or gene, which is known. And if this is the case, then that gene or QTL can be deployed by marker-assisted back crossing through our trait augmentation pipeline using speed breeding or via forward breeding through the trait pipeline and into the breeding program in the form of an improved parent. Where large effect QTL are not known, we can also look for trait variation within our pre-breeding material and use either genomic or phenotypic assisted pre-breeding and take forward the germplasm itself into the population improvement phases. And this goes all the way through to the point where we have no large effect QTL for traits of interest such as yield, but we have populations amenable to genomic selection. And here we're really looking at the large scale deployment of genomic selection for the advancement of material in the early stages of yield testing, as well as for the rapid recycling of parents to accelerate this breeding cycle. And the supply of this trait variation is not a current limitation. This is an example of a catalogue of QTLs which have been identified in CIMMYT projects in CIMMYT germplasm. And this shows you that we really have this abundance of supply of QTLs, of marker trait associations. And really the challenge now is to work out how to validate these and then to deploy them in the breeding program. And beyond QTLs, we know there's a lot of interesting variation that is hidden underneath these QTLs in the forms of haplotypes. And now with the growing body of sequence data available, we're really looking at how we move from QTLs to haplotypes and use this to make better selection decisions. And much of this work is in collaboration with Christopher Wowy's group at the John Innes Center as part of the new CIMMYT John Innes Center strategic collaboration on wheat research. And we aim to take forward some of these methods developed at John Innes Center and really look at how we apply them within the breeding context. And this is also synthesized in Philemon Juliana's talk later in this session. And Philemon will be talking about the data-driven approach to breeding, where we're using multiple sources of data to make selection decisions. And this is work that is currently underway, and we can look at all of the different stages of the breeding program, from the small plots to the third stage yield trials. And then Philemon will outline the application of different technologies, different data-driven approaches to move material through selection advancement and out of the program and into the hands of partners. And this is really a large-scale effort to apply these latest methods and data-driven approaches to accelerate the breeding process. And our yield gains are also in the context of the nutritional benefits that can potentially be derived from wheat as a staple food source. And the work that Velu Govindan's been doing on zinc mainstreaming, as well as specifically targeted zinc breeding, is represented here. And in Velu's zinc breeding pipeline, which is specifically selecting for both yield and grain zinc, you can see here a recent analysis of annual yield gains of 0.75% over time. So here we're showing grain yield progress, whilst at the same time, there's an average increase of 6 parts per million in grain zinc. So this is an exciting result because it indicates that we can co-select on a quantitative basis for both yield, obviously one of the major selection criteria, as well as grain zinc in the efforts to increase the levels of grain zinc via fortification and to address malnutrition and hidden hunger. And further work is ongoing with this material as well, because we're also interested in what happens to grain zinc in the processing stages, particularly in chapatis and bread, typically made from wheat flour. And this is work done by Itria Eber in the cereal chemistry labs here at Zimmit, and which really shows an impacts of extraction rate, the extraction of the flour having an effect on the zinc availability, but that the bread and chapati production itself does not negatively affect a zinc concentration in the final product. So this is really helping us to contextualize these increases in grain zinc and understand really what they can offer to the human nutrition perspective. So I've talked so far about securing supply, and just to summarize where we are on the supply side, as I mentioned before, we have over 50 years of delivery of improved germplasm with global impacts. We're applying new breeding methods to accelerate the early breeding generations. We're using methods to rapidly accumulate and incorporate research innovations into breeding, including this biofortification target to address hidden hunger. We're also developing closer CG-NAS networks to make material available faster and in a more targeted way to the geographies which we serve. But now I wanted to switch to the demand side, which is potentially had less attention in the context of the CIMMYT program and many public programs over time. And I guess here we're more asking questions at this stage and really thinking about what impacts we could have by also addressing the demand side of the equation. The first of these is addressing supply the only option to tackle the persistent issues of low uptake and slow varietal turnover? Is it simply a question of making better varieties and getting them to people faster? Is that the answer to varietal turnover, or does that miss some of the drivers of uptake and adoption? The second question is where the public sector is responsible for variety release, as we know is the case of wheat. Can we do more? And what are the incentives and models for the private sectors to become more engaged, particularly in the geographies where CIMMYT works? We also ask, how do we properly address preferences, markets, and gender beyond thinking of our trait packages and our product profiles from a breeding perspective in terms of the maturity type or the grain color? Can we understand a bit more about why farmers prefer certain varieties and why they choose to grow them or buy them, et cetera, et cetera? And finally, how do we design seed system interventions that can scale? Because this is really what we are about having solutions that scale, that can reach the maximum number of people and have the most impact on the supply of seed of improved varieties. And this work is led by the socioeconomics team here at CIMMYT, Jason Donovan and Peter Rustat and Rachel Voss. And this is really addressing some of the critical information gaps in how we understand farmer preference. So stakeholder preferences and needs need to be fully understood, both now and in the future. And we need to design systems whereby we can capture these preferences and really translate them into our product profiles or our breeding objectives. We also know not enough about the gender relationships in farming systems, who makes decisions on inputs and who has access to resources at the household level. We also need to understand far more about farmer decision-making around seed. What incentivizes the purchasing of seed? What's the role that risk plays? And what's the influence of the price or the information we provide alongside a bag of seed? And so in summarizing the next steps for this, we need to improve the study of preference by capturing farmer feedback in the household context, because individual farmers don't exist in isolation, but rather as members of households and communities which make decisions based on the influences of those around them. We also believe that we need to move beyond harvest stage evaluations, so the simple traditional approaches to participatory variety selection, where we're evaluating something simply at a single stage at the harvest point. And we think that differences in preference are likely to go beyond gender, and that market segmentation, which gives us greater clarity and greater information about what markets are really driving demand, may provide clearer answers. So who are the different types of farmers we seek to reach, and which of these groups are disproportionately made up of women? So this greater granularity of data, we believe, will allow us to understand much more about preferences and what's driving the demand for improved wheat varieties, for improved cereal seed. And a lot of this work builds on work done in the maize program and understanding some of those drivers of demand. Obviously, maize is quite a different system, but we're really asking the question, what can we learn from that work? So we know in maize, participatory varietal selection is quite widely used, and this uses large-scale variety trials with farmers, men and women, invited to evaluate varieties at harvest. So here you can see some farmers come and assess the material and make observations of it in the field. This is really a useful tool for researchers and breeders to determine which of the traits are of interest, and therefore informing which traits they should evaluate. And it potentially saves funds and effort by using these varietal evaluation trials, which would exist anyway, to collect an additional layer of data. So they're a relatively low-cost option for the breeding programs to solicit evaluation, but they occur really at the final stage of the breeding process. When a decision is made that this is a candidate, it will enter this participatory varietal selection process. So the takeaways from the work that the socioeconomics team have been doing is that preferences are often inconsistent between studies. And this is not ideal, obviously, from the breeding context, because we want to really have tangible targets when we're trying to incorporate farmer preference. Interestingly, men and women farmers tend to demonstrate generally similar trait and varietal preferences. So again, here we're not really drilling down into the granularity of what drives the gender dimension of crop production. And many of these studies suggest that women care about a wider range of traits, but the take home is that this doesn't necessarily mean that these traits are more important, nor that they'll directly impact on the choice of variety that a female farmer will make. And extending on from this into the seed system, there are also many unanswered questions, which are really the focus of the work we plan to do within the 1CG Seed Systems Initiative. And here we're asking, do gender gaps exist in wheat seed use? And if so, what is the primary driver of this? And much of the evidence points towards access to resources, which is obviously a huge factor and a huge consideration when designing interventions into the wheat seed system. And how to access to all of the things which are underpinned by resources, labor, land, information, education, and physical cash influence seed use, and what are the implications for women. And Rachel Voss, who's leading this work, is designing sets of experiments to really say, can understanding these components provide us tangible insights into how a female farmer would choose a variety? And if so, then can we incorporate this back into the design of those varieties, as well as into the marketing and dissemination of the varieties themselves? So in terms of improving seed systems access, there are 3 focus areas under the AGG maize component, which we hope to extend to wheat within the 1CGIAR Seed Systems Initiative, which is called Seed Equal. And Seed Equal really highlights the need for equal access to seed for all, as well as equitable access to quality seeds. The first is about the gender dynamics of plot management. So a need for the understanding of women's roles in plot management and decision-making. Should breeding tailor seeds to unique preferences? I think this is a question which is still outstanding, and also which informs the breeding process. To what extent can we market seeds directly to women? We know many of the interventions now used for marketing are in fact exclude female farmers from the process. And do we need to confront other underlying barriers to seed access? And do we have a way to actually do that? The second is on product concept testing. And this addresses the challenge that our current product profiles are largely based on our existing breeding pipelines. We know how to get varieties registered, and we work backwards from these criteria to define our breeding product profiles. But here we ask the question, can we use social science and an understanding of preference and farmer demand to bring better data on preferences to the table, and thereby to the breeding program design? And then can we use the data collection methods to assess preference based on product description? So can we make real or pretend products and use these to evaluate preference in an effective and scalable way, engaging with the breeding teams and the breeding partners in design? And then the final aspect of this is agribusiness and the role of the private sector largely in seed markets in terms of the launch of new varieties. And this is about mapping the current seed offer to understand diversity and choice and changes in preference over time. So this is a very important component, because what we're essentially trying to do is balance the supply side with the demand side, because we believe delivering genetic gain into the breeding program is very useful, but ultimately we need to deliver this genetic gain into farmers' fields in the form of improved seeds, which are adopted and widely grown by farmers. So thank you very much for your attention. You can find out more about both the CIMMYT Global Wheat Program, our donors and our partners, as well as the Accelerating Genetic Gains in Wheat project via these links here. Thank you very much.